Today, I'm going to introduce our work on generalized Pearson correlation squares for capturing mixtures of bivariate linear dependencies. This is my joint work with Dr. Xin Tong at University of Southern California and Dr. Peter Bickel at UC Berkeley. Our motivation was from a paper published in 2011 in Science. In this paper, the authors proposed the maximal information coefficient as a novel association measure for capturing relationships between two real-value random variables. So in this paper, this table captured my attention. The proposed measure MIC was designed to be versatile, so it can capture relationships ranging from linear to parabolic to exponential to cubic. So the goal is that when there is no random noise, that is the data points lie perfectly in such a pattern, the MIC would return a value of 1. In the competitors, I saw the maximal correlation, which was designed to be versatile as well. So its definition is the maximal absolute Pearson correlation for fx and gy. So f and g are two function transformations of x and y. So by design, the maximal correlation should give a perfect value to these relationships as well. However, it doesn't. And interestingly, later I found that it was due to the lack of convergence, a computational issue. But still, I was wondering, what cannot maximal correlation do? In this paper, they also show that MIC can capture those non-functional patterns, which means that there's not one-to-one relationship between X and Y, such as the cross. Then the question is, are these non-functional patterns scientifically interesting and important? I came across this data set in graduate school from a classmate who worked on Arabidopsis data. This is gene expression data. And if we plot two genes in the scatter plot, we can see that their relationship actually depends on the tissue. In the root tissue and the shoot tissue, we see that the linear relationships may be different. If we ignore the tissue, we just look at all the data points together, we may miss some strong linear relationships existing within tissue. So motivated by this example, we consider a more extreme scenario. So in this case, what if the linear relationships within tissues are strong, but together the relationships cancel each other out? So if we look at all the data points together, we will see zero correlation. This is called Simpson's paradox in statistics. And the famous example is the Berkeley graduate admission, where we would have different conclusions about whether gender bias exists by looking at all the departments together versus each department at a time. So how do we do about this relationship? There exists a large body of work about scalar value association measures, and I categorize them into two types: functional measures and general dependence measures. So for those functional measures, they are designed to capture a functional relationship between two real value random variables. The simplest one is the Pearson correlation, and the more general ones were recently developed, and we can see that they capture more and more flexible and more types of functional relationships. On the other hand, the dependence measures care about anything that's not independent. So it's not restricted to functional relationships. It's worth noting that the maximal correlation is a special thing. It can satisfy both. It's designed to capture functional relationships, but it also has a special property. That is, its value is 0 if and only if X and Y are independent. So given this categorization, we can say that from linear relationship to general dependence relationship, the measures lose the interpretability but gains the flexibility. The Pearson correlation is the most interpretable but not flexible. Okay, So now, for this special type of measure we are interested in, the mixture of linear dependencies, it has the interpretation because each component is linear, but together it's not a functional relationship. So our question is, can we have a measure that specifically targets this relationship so that it's more powerful than general dependence measures? And it also has the interpretability as the Pearson correlation has. So regarding this mixture of linear regressions, it has been developed over 40 years across multiple disciplines. And existing work focused on algorithm development to find the lines and statistical inference for parameter estimation. However, they do not propose an association measure that can give a value for the pattern. And that's our question of interest here. So we're revisiting this old problem. We consider two scenarios, specified and unspecified. The specified scenario is the case where it's easier for us. We have an index variable so that the variable tells us which line each data point belongs to. The unspecified scenario is more common where we see the pattern, but we don't know where are the lines. So we want to use the specified scenario as a starting point to give us a foundation for investigating an unspecified scenario. And we want to answer the following questions. Under the unspecified scenario, can we simply fit a mixture of linear regressions and solve the problem? The answer is no, because linear regression does not treat X and Y exchangeable. When there are more than one component, we would actually, if we fit a mixture of linear regression, we will actually get different lines, and also the overall goodness of fit measure would also be not symmetric between X and Y. The second question is how to assign observations to lines to ensure the exchangeability of X and Y without distributional assumptions. We don't want distributional assumptions because biological data are so complex. We cannot just assume one distribution that works for all. So for this purpose, we propose a model-free k-lines clustering algorithm, which was motivated by the k-means clustering algorithm. The second question is, how do we define a population level measure for proper inference? We would ultimately, or ideally, we would desire a measure that works for both scenarios. However, we realize that without distributional assumptions, that's not achievable. We need to have a different population measure for the unspecified scenario from that under the specified scenario. The last question is, when an index variable is available, should we always choose the specified scenario? The answer is actually no, because the index variable could be uninformative or irrelevant to the segregation of lines. Here's an example of an uninformative index variable. Still from the Arabidopsis data, we see that the condition variable cannot really segregate the lines. So in this case, we would actually prefer the unspecified scenario because it will allow us to take an unsupervised and data-driven approach to find the pattern. As an overview, to address this question, we proposed 4 measures called Generalized Pearson correlation squares. 2 for the specified scenario, 2 for the unspecified scenario. Under each scenario, we have the population level measure and sample level measure. So to explain the notation, we use rho square for population level, R square for sample level, G stands for generalized, and S stands for specified, U stands for unspecified. We start with the specified population level measure. Our idea came from generalizing the Pearson correlation square. And it is natural to start with the conditional Pearson correlation within the kth line. So if we squared it, right, the idea is to remove the sign because we don't want to have the opposite signs cancel each other. Instead, we want to capture the overall strength. So we square the conditional correlations, and then we have this index variable z. Then we take the expectation for this index variable, and that's a natural definition for the specified correlation square. Then this can actually be written out as a weighted sum, where the weights are the probabilities of lines and is the weighted sum of the conditional correlation squares. So given this, we actually want to offer some insight into the lines of the memberships. So we want to offer an interpretation. What should be the line center in this case? Given that we desire x and y to be symmetric, a reasonable distance metric, distance between a point and a line, should be the perpendicular distance that can treat x and y symmetric. With this distance and with the specification, then we can define the specified population level line center as this. So it is the line that minimizes the expected squared perpendicular distance between x and y to it. So we can do this for every component, then we can have k lines in total. So how about the sample level? This is easy. Given our definition of the population level, we can do the plug-in, right? We can plug in the sample level conditional correlation squares and the observed line proportions. That will give us the sample level measure. Now we concluded the specified scenario and we move on to the unspecified scenario. Here, we only have two variables, x and y, no more z. How should we define the population level line centers? We still want to use the squared perpendicular distance and minimize it, but we don't have the specification. So our idea is that for any set of k lines, we can find the closest line to x and y, so in terms of the perpendicular distance, and we take expectation for x and y. This becomes our objective. We want to find a set that can minimize this objective, and that set will be defined as our k line centers in the unspecified scenario. And we borrow this idea from the population level k-means clustering. Okay, I want to say that the two sets of line centers, one under specified scenario, one under unspecified scenario, are different. This is by our definition. Without distributional assumptions, we think our definition is a reasonable definition for the unspecified line centers because it will encourage the line to be closest to data points. So it is not surprising that here we will have a larger per line conditional populations Pearson correlation square than in the specified case. Okay? And given the lines, we can define this random surrogate index, which depends on x and y and the k lines. It is the index of the line that is closest to x and y. Given that x and y is random, this index is also random. It's induced by the joint distribution of x and y. Okay, with this z tilde, now we can define the unspecified population level measure. So the idea follows from the specified scenario. We just replaced z by z tilde. And we can prove that this population level measure in the unspecified scenario is greater or equal than that under the specified scenario. And how do we define the line centers in the sample case, sample level? So we follow the idea in the population level, we just replace that expectation to be minimized by average. And the idea is the same. Given a set of k lines, for data point, ith data point, we find the closest line, calculate the perpendicular distance, squared it, and take the average over data points, and we find the set of k lines to minimize this average. That will give us k lines. And to find those k lines, we propose a k lines clustering algorithm. So with the k lines, now we can define the sample level surrogate index, one per data point. It is the index of the ith data point, the line it is the index of the line that the I data point is closest to. We call this zi tilde hat. And this is actually obtained by the k-lines clustering algorithm. With this, now we can define the unspecified sample level measure. What we did is the same as in the specified scenario, but we replace zi by this zi tilde hat. Now we finish the definitions. But a practical issue is how do we choose k in the unspecified scenario? We offer two practical solutions. The first is that we can use this common criteria, average within cluster sum of distances. Here, we use the perpendicular distance because our cluster center is a line, and we use this distance for symmetry, which is also consistent with our objective. And we can use the elbow plot to choose a proper k. Or if we are willing to assume data distributions, we can use this model-based AIC criteria to choose k. So to enable inference, we derive the asymptotic distribution for the sample level measures. Under the specified scenario, the derivation is not difficult. We can just employ the delta method to derive it, and the variance, if we want to assume the simple case of a mixture of bivariate normal distribution, the variance has a clean formula. It only depends on the line probabilities and the per line conditional Pearson correlation squares. For the unspecified scenario, we need strong consistency of the k-lines clustering. In other words, we need the sample level k-lines to converge to the population level k-lines, almost surely. So with that, we can derive the asymptotic distribution of the unspecified sample level measure. And it also can allow us to do the inference for the population level unspecified measure. All right. We will show the simulation result to demonstrate the power of the proposed approach. So here we start with a simple linear and parabola patterns, and we can see that among the measures, including Pearson correlation square, maximal correlation, distance correlation, MIC, and our unspecified measure, we can see that they're all doing very well in the linear case. In the parabola case, the Pearson correlation square does not work well as it's supposed to be. In the pattern we're interested in, the noiseless pattern, which is a cross, we can see that our measure clearly has the desired advantage, especially when the sample size is small, like 30. We also look at this more complicated pattern, which can be approximated by lines. So I designed this pattern to make it more like a real data case. So even in this case, which is not linear but can be approximated by linear, our pattern still has clear advantage over existing measures. Now we see some real data application. The first application is the motivating example. And we have two groups of gene pairs. One is the pairs of genes in the GSL pathway, which means that these genes are known to be biologically related. Another group contains random gene pairs. So scientifically, we would expect the gene pairs in the first group have overall higher associations than the genes in the second group. Is that the case? We can see that the Pearson correlation doesn't show this. Instead, it shows that the random gene pairs have an overall higher measures, which is not reasonable. Maximal correlation, distance correlation, MIC all suggest the same pattern, as well as the uninformative index variables, condition, treatment, replicate. If we use them in the specified scenario, we got almost the same pattern as the Pearson correlation square. Tissue is the only exception, as we already saw. It is an informative index variable. With it, the specified generalized Pearson correlation square give us higher association among the GSL gene pairs. That's aligned with our scientific knowledge. And our unspecified measure, so it's doing a very good job. If we see it, it actually shows the GSL gene pairs have stronger associations than the random gene pairs. And it is the only one that shows this pattern among all the unspecified measures, including R-squared, max-core, D-core, and MSE. The second application is about the rising single-cell technology data. So in this data, we have cells which may come from different types. Therefore, it is reasonable to expect that the gene-gene correlations may exhibit mixture of linear relationships. So to find interesting gene pairs, we would expect a bimodal distribution so that those high values would indicate interesting pairs, low values otherwise. And our unspecified measure is the only one that gives the bimodal distribution across gene pairs. But maximal correlation has a weak bimodal distribution, but not as obvious as ours. And the R-squared shows no bimodal distribution at all. Okay, here's the summary. We proposed a set of generalized Pearson correlation squares. They include two scenarios, specified and unspecified, population level measure and sample level measure. And we enabled statistical inference by deriving the asymptotic distributions of the sample level measures. We proposed a K-lines algorithm for the unspecified sample level scenario to derive the sample level measure. And future applications, we already showed that this measure will be useful for studying gene expression data, and we expect it to be applied to other gene expression data analysis. And another important application we have in mind is the genetics, the genotype-phenotype association studies. So we expect that our unspecified measure can capture hidden human subpopulations, which we don't know beforehand. This is the archive version of our manuscript, and this is the R package that implements the algorithm for computing the measures. And finally, I want to thank my collaborator, Dr. Xin Tong at University of Southern California, and my former PhD advisor, Dr. Peter Bickel at UC Berkeley, and the funding agencies for supporting my research. Thank you.